AIDS was first reported in the year 1981, and in the last 25 years or so, it has spread all over the world, killing more than 25 million people. And there is always a time lag between the infection and the appearance of AIDS symptoms. There is no particular treatment for AIDS. Only thing is the symptoms or the lifespan of the individual can be increased for certain years. Other than that, AIDS cannot be completely cured because it is a viral infection. Hello everyone, a warm welcome to the session for class 12 biology. I'm Dr. Divya, biology faculty. So in this session, that is session 6 of chapter 7, human health and diseases, we shall learn about the diseases of human, wherein in this session particularly we'll talk about the acquired immunodeficiency syndrome, that is AIDS. So AIDS, the full form of AIDS, it is very important in terms of MCQ. So AIDS is acquired immunodeficiency syndrome. So we can learn about it by splitting the words. So when I say or learning the meaning of each of these words, when I say acquired means we get it from somewhere, right? So AIDS doesn't automatically develop in our body. It's not an inborn disease that we have, but we get it from outside because of a particular virus which is called as the HIV. So therefore we acquire it. So we get it and spreads from one person to another. So we acquire the virus from one person, from an infected person. So therefore the word acquired, it's called immunodeficiency. The word itself says immuno means it is something related to the immune cells in a body, be it the lymphocytes, that is the B lymphocytes and the T lymphocytes or the WBC as such. So there is a deficiency in the immune system. Why? Because this particular disease which we acquire from another person due to the infection by the retrovirus, that is a HIV, our immune cells will, the count in the immune cells will become lesser. So therefore we will be deficient of immune cells in our body. So that is why the name acquired immunodeficiency. Now talking about the word syndrome. Syndrome means it is a collection of diseases, because once we get AIDS or once a person is infected with AIDS, then the immune cells will drop down. So therefore, if the immunity is very, very weakened in the body, then we can get a number of other diseases. So therefore, AIDS is acquired immunodeficiency syndrome, wherein it results in the deficiency of the immune system and it is acquired during the lifetime of an individual, and therefore it is not a congenital disease. So what is congenital disease? Congenital disease means it will be there right from the beginning, but in this case it is not so, right? We get it from outside. And the syndrome meaning group of symptoms. So AIDS was first reported in the year 1981, and in the last 25 years or so, it has spread all over the world, killing more than 25 million people. One thing is, it is very difficult to cure, and also people will not take proper measures to prevent these diseases. So what are the measures we need to take, how to control, and all that we'll study in the coming slides of this session. So this is about AIDS. And AIDS, the causative organ, the causative organism of AIDS. So it is caused by a virus which is called as human immunodeficiency virus, that is HIV. And HIV belongs to a class of viruses which is called as retrovirus. And these retroviruses, they have a protective envelope or a covering, and inside the protective covering, so we know that all the viruses, they have a protein coat outside, right? That is a protective envelope, or we call it as the capsid. So within that protective envelope, they have RNA as the genetic material. And we know that viruses, they can either have DNA as the genetic material or RNA as the genetic material, and both DNA or RNA as the genetic material in them. So in this particular case, the HIV, which belongs to a group of retroviruses, they have RNA as their genetic material. So that is about the HIV. So we got to know about what is AIDS, we got to know about the causative organism, that is human immunodeficiency virus, and what is it made up of? It has a protective coat. Inside the protective coat, what is present? Genetic material is present. What genetic material? It is the ribonucleic acid or the RNA. Now talking about the transmission of HIV infection. So we all have a myth, right? Like whenever we get to know a person is infected by HIV or by AIDS, we think that by just touching that person, we can get the infection, or just by having a meal, sitting and having a meal with that person, we can get the infection. It is not so. So we will just have a clarity like exactly how this particular HIV infection generally transmits from one person to another. The first main reason is sexual contact with the infected person. And next reason is transfusion of contaminated blood and blood products. So whenever we fall sick or we have a surgery or something, we need blood transfusion, right? During that time, the donor's blood need to be checked properly for HIV, wherein ELISA test and all that can be performed. And if it is only free from HIV, then the blood needs to be taken by the blood bank. Otherwise, the blood should not be accepted from the donor. So because of the negligible or because of these tests being neglected, HIV can spread through blood transfusion also. That is by blood transfusion of contaminated blood by the HIV virus. And one more thing is sharing of infected needles. So nowadays, earlier what they used to do is they used to sterilize the needle and use it again and again, right? But nowadays we have needles or syringes which are of one-time use, that is disposable ones, right? So earlier it was not like that. So sharing the syringes in the hospitals with one patient to another, and if one patient is infected with HIV, then the disease can spread to the other patient who has been injected with the same syringe, right? So it can spread. Not just that, HIV is more common among the drug addicts. Why? Because some of the drug addicts who sit in a group and share the syringes to inject the drug into their veins, they also can get infected. So in that one person, if he has an HIV, then the infection can spread to the whole group who has used the same syringe. So it can spread by sharing infected needles, as in the case of intravenous drug abusers. And not just that, it can also spread from the mother to her child through placenta. Because we know that all the nutrients from the mother is supplied to the developing fetus inside her through the placenta. Similarly, if the mother is infected by HIV, the HIV virus can enter the fetus or the baby through the placenta also. But this can be prevented by early diagnosis. So that is why every pregnant woman at certain time, when she reaches about 3 months or so, a ELISA test will be conducted, which is compulsory, or a HIV test will be conducted, so that early detection can protect the baby from HIV. So these are the causes, like how HIV transmits from one person to another. So apart from this, by shaking hands with the HIV person, by sitting with the HIV person, or if the HIV person's saliva splashes on us and all that, we will not get HIV. So HIV can be transmitted only through those 4 ways which has been mentioned in the previous slide. So next we'll look into who are the people who are at the high risk of getting infected by the HIV virus, therefore falling into the prey of AIDS. So the individuals who have multiple sexual partners, they can be infected. They are more prone to infection by the HIV person. And people who take drugs are more, that is especially intravenous drugs, wherein syringes are injected into the veins, they are more prone to infection. And individuals who get blood transfusion, that is repeated blood transfusion, where the blood banks don't check the blood of the donors and all that, and by mistake if they put that blood into the recipient, then they can get the HIV infection. And also children born to an HIV infected mother. So earlier there were no measures that could be taken, but nowadays early detection can help protect the fetus from HIV. So these are the people who are at the high risk of getting HIV. So if we avoid all these, then we are in safe hands. Next, talking about how HIV doesn't spread from one person to another. Knowing this is also very, very important, because we always look upon those people infected by HIV as people who should be kept aside from the society, with whom we should not mingle, right? So it is always good to know like how HIV doesn't spread. So you have already got to know how HIV spreads from one person to another, and who are at a higher risk of getting HIV. Now we'll learn how HIV doesn't spread from one person to another. So HIV is not spread by mere touch or physical contact with the infected person, and it can spread only through body fluids, that is through semen or through blood transfusion or through intravenous injection or from mother to the child through the placenta, that is the fluids that pass through the placenta to the fetus. So that is the only way it can spread, not by touching or keeping in contact with the infected person. And therefore, for the physical and physiological well-being of the HIV infected people, the HIV infected people should not be isolated from the family and the society. Some of the people, they on purpose, they get this particular infection because of the bad habits that they have. But some people unknowingly, because of the blood transfusions and all that, they can get the infection. So we have to have a respect towards them and not keep them away from the society, because now we got to know that it is not spread just by touching that person or mingling with that particular person, and it spreads only through body fluids. And there is always a time lag between the infection and the appearance of AIDS symptoms. So HIV, why we cannot detect it at an early stage? Why? Because the main reason is the AIDS, almost to show up the symptoms, it may take months or to many years, so 5 to 10 years, and that person will not know that since 5 to 10 years he is having this particular HIV virus in the body, because it is slowly infecting without causing any major symptoms. So that is the reason the person will not get to know, and by the time it is being detected, a lot of damage would have already been done to the body. So that is one of the reasons why HIV early detection is not that early detection is not that possible with the people who have that infection. So this is about the AIDS, so how it does not spread, and also the symptoms to appear in AIDS, it takes months together, or sometimes it can take 5 to 10 years also before it is being detected. So next, talking about the infection process, that is how the infected cell. So this is a very important question which was asked last year on the question paper also. So this question can be asked as, give the schematic representation of replication of retrovirus or replication of HIV in particular. So if that is the case, we have to write this schematic diagram or HIV. We have to write this schematic diagram, or sometimes if we look into the 2024 question paper, the question was framed, that is by the Karnataka PU board, the question was framed in such a way, how can an infected cell or show the schematic representation to depict an infected cell can survive while viruses are being replicated and released. So this is how the question can be framed and asked. So they can give the question as, give the schematic representation of replication of retrovirus, or give the schematic representation to show how an infected cell can survive while viruses are being replicated and released. So for both these, this is the schematic diagram that you need to, or schematic representation that you need to put in your answer booklet if the questions are asked. So talking about the replication of retrovirus inside the human body or inside the cells. So retrovirus, so HIV belongs to a class of retrovirus, and what is their body made up of? It has a protective envelope which is made up of protein. So therefore, it has a viral protein coat, and inside the viral protein coat is present the genetic material which we call as the RNA, which forms its viral genome. So RNA is the genome, or RNA is the genetic material present inside that. So these viruses, they infect the normal cell. Say for example, animal cell. We human beings are a best example. So when they infect the animal cell, what they will do? They will transfer the viral RNA into the cytoplasm of the animal cell. So what they will do? They'll transfer the viral. So this is the animal cell. This whole portion, it is what they have shown. No, it is the animal cell. So what did the virus do? It embedded itself onto the plasma membrane of the animal cell. It embedded or sticked itself onto the plasma membrane of the animal cell. Once it got embedded in the plasma membrane of the animal cell, it released its viral DNA which is produced by reverse transcriptase. Now, what is the it's written as viral DNA produced by reverse transcriptase. So, viral RNA is introduced into the cell. That viral RNA gets converted into viral DNA. How the conversion happens? Because of the presence of an enzyme which is called as reverse transcriptase enzyme. The viral RNA gets converted into a viral DNA. Now this is the viral DNA. So this viral DNA gets incorporated along with the DNA of the host genome. So host genome means inside the animal cell. And therefore, next, new viruses will start to get produced. New viruses will start to get produced, and these new viruses which are produced, they can move on to the new cells. So here also cells will be there like this. Wherever new cells are there, they can move on to there and they can spread the infection. So that is how they survive. This is how the retroviruses survive inside an infected cell and spreading into new cells causing infection. Or this is how the replication of retrovirus or HIV happens inside a human cell or an animal cell. So what happens? You have to remember this. First it will deliver the viral RNA into the cytoplasm. Then the viral RNA gets converted into a DNA with the help of a reverse transcriptase enzyme. You have learned this in molecular biology chapter. And that viral DNA will go and incorporate itself along with the hosts or the animal cells DNA that is present inside the nucleus. That DNA will later on, it will start producing into, when the cells divide, new viruses will start form, or daughter viruses will start forming. Those viruses can infect the neighboring cells which are there. So this is the life cycle of the replication of the retrovirus or the HIV virus. Next, talking about the symptoms of AIDS. So symptoms of AIDS, as I said, it takes about months or 5 to 10 years for an infected person to show symptoms. So what are the symptoms of AIDS? The person suffers from bouts of, that is, frequent fever, diarrhea and weight loss. And due to the decreased number of the immune cells, especially the helper T lymphocytes, the person starts suffering from infections that could have been otherwise overcome, such as those due to bacteria, especially mycobacterium, viruses, fungi, and even parasites like toxoplasma. So all these viruses, fungi or bacteria or the parasites are killed inside a body by the immune cells which are there. So now what happens in a virus infected person, especially the HIV infected person, his immune cells count will go down, right? So therefore the helper T lymphocytes which are there will fall down drastically. So therefore his body will be weakened without a proper immune system. So therefore is prone to a lot of other infections by the bacteria, especially the mycobacterium, various viruses, fungi and parasites like the toxoplasma. So therefore that is why we call it as a syndrome, a collection of diseases. And the patient becomes so deficient in his immune cells that he or she will be unable to protect himself or herself from these infections. So any small infection can cause a lot of problems in the person whose immune cells are very, very weakened. The best example is in a person who is infected by HIV. Now, what is the diagnosis and treatment for HIV? So, talking about the diagnosis, so there is a test which is called as ELISA, that is enzyme-linked immunosorbent assay. So, this ELISA test is used in the detection of the HIV present. So, it detects the number of antibodies that are formed in a cell. So, antibody which is particular against the HIV virus. So, more number of the antibodies pertaining to the HIV virus is formed in the blood, then it is understood that, okay, the person is infected by this particular disease. So, that is how it detects the HIV in the person's blood samples. That is called as enzyme-linked immunosorbent assay. And talking about the treatment of AIDS, the treatment, there is no particular treatment for AIDS. Only thing is the symptoms or the lifespan of the individual can be increased for certain years. Other than that, AIDS cannot be completely cured because it is a viral infection. So the treatment of AIDS is done using certain antiretroviral drugs And also with the use of antiretroviral drugs, the patient's lifespan can be prevented. But death can happen, but maybe the lifespan can be increased up to certain years. So this is about the diagnosis and treatment. So diagnosis is done by a tool which is called as ELISA, and treatment is by using antiretroviral drugs. Now, how to prevent AIDS? So, prevention is always better than cure, because once a person is infected by AIDS, there is no cure for it, right? So, therefore, always it is better to prevent ourselves from getting AIDS. So, National AIDS Control Organization, which is called as NACO, and other non-governmental organization or NGOs, they have been doing a lot of work or contributing a lot to the society in terms of educating people about AIDS. So in villages and all, a lot of banners will be put up, or even whenever you go to a movie theater also, a lot of advertisements will be put up before the movie starts, or when you go to a bus stand or somewhere, or to a transport stand, you can hear announcements about AIDS and all that. So all these, or in the newspapers and the advertisements, everywhere a lot of awareness is being created to prevent AIDS. And the World Health Organization has started a number of programs in order to prevent the spreading of the HIV infection. And also, it is made sure that the blood banks are safe from HIV. So, any donor who is donating the blood needs to undergo ELISA test to check for HIV infection before the blood is collected by the blood bank. So, therefore, blood bank should work together to check the donor's blood for infection. And also, only disposable needles and syringes should be used in public and private hospitals and clinics. And also, free distribution of condoms need to be done. There are some states, even in India also, wherein there are ATMs, like how in ATM you can draw, that is money from the bank. Similarly, there are ATMs which act like, so how you have that vending machines wherein you'll put a coin and you'll get the product that you want, right? Same way, there are vending machines for condoms wherein you can just put a coin and then you can take it, because some people, they'll feel shy to ask for it in the public, right? So therefore, all these facilities are being given or done in order to prevent or control AIDS. And controlling drug abuse and educating, especially the adolescents, educating them about the intravenous drug abuse and all that can lead to prevention of AIDS. And advocating safe sex and promoting regular checkups for HIV in susceptible population, so that HIV spread can be prevented. So these are the preventive measures that can be taken in order to prevent the spread of AIDS. So this was about this session wherein you learnt about the dreadful disease which is called as AIDS, or a syndrome which is called as AIDS, the pathogen that causes it, how it spreads from one person to another, and how the virus spreads inside the body of the infected person, and what are the preventive measures for AIDS, all that we have seen. So in the coming session, we'll talk about one more dreadful disease which is called as cancer, and we'll learn about the technologies that are used for detecting and diagnosing cancer. So see you in the next session with these two topics. Thank you.